This lecture, part 2 of 4, will cover the structure and function of cells. The topics of filtration, carrier-mediated transport, and vesicular transport will be covered. Filtration is a passive process where hydrostatic pressure forces water across a membrane. Hydrostatic pressure is created when a force is applied against a solution. If a solute molecule is small enough to fit through pores in the membrane, it will be carried along with the water. An example of filtration in the body occurs at the kidneys. Blood pressure against specialized vessels found here results in filtration, an important process in the formation of urine. Both carrier-mediated transport and vesicular transport assist in the passage of compounds across the plasma membrane. Carrier-mediated transport requires membrane proteins and can be passive, with no energy being required, or active, where energy from ATP is required. The membrane proteins bind specific ions or substrates that require aid in the passage across the plasma membrane. This can be done through several different mechanisms. In co-transport, two substances are moved across the membrane at the same time and in the same direction. So co-transport, two substances, both going in the same direction. With counter-transport, on the other hand, two substances are again moved across the membrane, but this time in opposite directions. Counter-transport results in the movement of two substances in opposite directions. In other words, one gets in and one gets out. Also, substances can be moved one at a time by carrier proteins. Facilitated diffusion is a specific type of carrier-mediated transport that uses carrier proteins to passively move large compounds down their concentration gradients. Since this is a passive process, energy is not required. The process works by a compound binding to the receptor site on a carrier protein. The shape of the carrier protein then changes, moving the compound across the membrane. Unlike simple diffusion, the rate of facilitated diffusion is limited by the number of carriers found in the plasma membrane. This diagram demonstrates the passive process of facilitated diffusion. Here the substance being examined is glucose. The concentration of glucose is higher outside the cell compared to inside of the cell. Therefore, to flow down its concentration gradient, the glucose will want to travel into the cell from the higher to the lower concentration. The glucose molecule will be able to bind to the glucose receptor site on the carrier protein. When this occurs, the protein will change shape, allowing the glucose to pass into the cell. Active transport, unlike passive transport, requires energy and is capable of moving substances against the concentration gradient, that is, from areas of lower concentration to areas of higher concentration. Ion pumps are found in all cells and carry essential ions to one side of a membrane from the other. Common ions that are transported by ion pumps include sodium ions, potassium ions, calcium ions, magnesium ions, as well as iodine ions, chloride ions, and iron ions in some cells. If the ion pump allows for one type of ion to enter the cell and another type to exit the cell at the same time, it is known as an exchange pump. An exchange pump is an example of an ATP-driven countertransport mechanism. A sodium-potassium exchange pump is a specific example, and the following diagram demonstrates how this pump works. So here we see the sodium-potassium exchange pump. The sodium-potassium exchange pump requires energy to run. Recall that ATP contains high-energy bonds that, once broken, will release energy for the cell to use. For every adenosine triphosphate or ATP molecule that is converted to ADP, enough energy will be released to allow for the pump to move 3 sodium ions out of the cell while at the same time moving 2 potassium ions into the cell. This again is an example of active countertransport. In vesicular transport will provide a means to move large amounts of materials into or out of the cells in small membranous sacs known as vesicles. There are 2 major categories of vesicular transport: endocytosis and exocytosis, and we will take a look at each. In endocytosis, extracellular material that's found outside of the cell, is packaged into vesicles at the cell surface and then moved into the cell, a process that requires energy. There are 3 key types of endocytosis: receptor-mediated endocytosis, which provides vesicles containing high concentrations of a specific molecule; penocytosis, which produces vesicles of extracellular fluid. This can be thought of as cell drinking. And finally, phagocytosis, which produces vesicles full of solid objects and can be thought of as cell eating. The process of receptor-mediated endocytosis is depicted here. First, target molecules known as ligands bond to receptors in the plasma membrane. So we're going to have all these ligands bonding to their receptors. Next, a deep pocket in the area the bond ligands forms. This pocket will be pinched off to create what is known as a vesicle. The vesicle enters the cell and then fuses with a lysosome, creating this complex seen here. The ligands are removed and released into the cytoplasm of the cell. The lysosome then detaches, and the empty vesicle with the ligand still present in its exterior will go ahead and travel back to the membrane of the cell, and it will go ahead and reincorporate itself into the plasma membrane. This again is an example of receptor-mediated endocytosis. In the process of phagocytosis, again, you can think of that as cell eating, a foreign object is surrounded by extensions of the cytoplasm known as pseudopodia. The pseudopodia connect and fuse, creating a vesicle around a foreign object. So here we have a foreign object on the outside of a cell, a phagocytic cell. These pseudopodia are going to come and form around that foreign object, eventually closing off and creating a vesicle, seen here. That is what is brought into the cell. Lysosomes then fuse with this vesicle, as seen here, and the digestive enzymes that are inside of the lysosome will come in and break down the foreign object. The nutrients are then absorbed, and any leftover residue is gone ahead and brought out to the outside of the cell in a process known as exocytosis, which we will examine next. Exocytosis is the reverse of endocytosis. In this process, material is moved out of the cell through the use of vesicles that merge with the plasma membrane. This process also requires cellular energy. Some common materials that exit the cells via exocytosis include waste products, mucus, and hormones. So again, if we go back to the previous slide, we can see that here, where this vesicle is being brought to the cell membrane and then its contents are being released into the extracellular fluid, that would be the example of exocytosis. Let's go ahead and review with a checkpoint. What is the difference between active and passive transport processes? Feel free to pause the lecture while you formulate your response. Active transport requires energy, while passive transport does not. Can you name some passive processes? Some passive processes that we discussed included diffusion, osmosis, filtration, and facilitated diffusion.